I'm Melissa Wilson-Sayers, and on behalf of the organizing committee, Erin Quinlan and Janet Kelso, I'm excited to introduce our second keynote speaker. I thought I would just give a very brief story of when I met Lior. So I first moved to Berkeley in 2011, and it was big and new and different. I came from small town Oklahoma and Nebraska, right? And Berkeley was very unknown. And I met this brilliant bioinformatician who was very welcoming and very witty and very smart. And she introduced me to her husband, Lior Pachter. And through that, then I was able to get to know his work. So Lior did his bachelor's in mathematics at Caltech, his PhD in applied mathematics at MIT, so going across the country, and then back to Berkeley for a postdoc, assistant associate, full professor, and then in January 2017 moved back to Caltech. And with that, I'll let you start your talk.
Very much. Thanks so much, Melissa. Thank you for inviting me, and thanks also to Janet and Erin for giving me the opportunity and the privilege to be at this meeting. It's really true that this is my favorite meeting, and I've said this before. I've met future collaborators here, had many ideas here, and learned a lot of things here always, so I'm really privileged and happy to be here. My talk today is about post-procrustean bioinformatics, and apparently in anticipation of my talk and seeing the title, people have been learning Greek mythology this week. But I'll tell the Procrustes story in a minute. First, I'd like to start by talking about raw data. This is key to the talk, actually. Raw data is a term we use all the time as bioinformaticians, like many scientists. And I think it's true that we like to imagine raw data is this beautiful thing. And to give some adjectives, it's clean, it's untarnished, it's juicy, it's fresh, and it's going to be really, really good. But the reality is that we don't always agree on what raw data is. Raw data is a very relative term. One person's compost is another person's raw data, is really the way it is. And so even though the idealized version of raw data looks something like that, in actual reality, at least in genomics, raw data for biologists means that you put your stuff on the short read archive. If you didn't do that, you didn't share your raw data, and you get scolded, and it's not okay. Statisticians, on the other hand, for them, raw data is usually something that looks like this. This is an example from single-cell RNA-Seq, which is something I'll talk about today a little bit. But generally, it's a table of numbers, counts that you put into R. And it's really true that if you look at, in preparation for this, I did some sleuthing about this. And if you look at most papers, the statisticians always say, I'm going to start with the raw data. They really mean this. In reality, raw data is a PDF of an Excel file. So, fortunately. So there's one example, and that's why the previous slide, my table was single-cell RNA-Seq, where basically everyone agrees on what the raw data is. That's not always the case, but in this case it is true. And it has to do with this new technology, 10x. It's a year or two old now, for single-cell RNA-Seq. And it's a great example where really there's complete consensus, and that's why I want to talk about it. So I want to briefly review the technology at a very high level. Both this characterization and the picture is all from 10x itself. This is from the 10x website. This is a figure from a paper 10x wrote, obviously advertising the technology last year. So what it means to do single-cell RNA-Seq is you're measuring expression of transcripts in individual cells. And the way 10x accomplishes this is essentially via a microfluidic system that is really quite simple in its structure. There really are three components: beads, cells, and oil. And the idea is that you're going to end up, that's what this picture shows here, with little droplets that are really an emulsion, which are aqueous droplets in oil, which encapsulate ideally one cell and one bead. And well, why are you trying to do this gymnastics is that the bead will have a sort of DNA barcode on it. So if you're inside one droplet now and you have just one cell with one bead, the mRNAs, which are in this cartoon here, will get captured. Sorry, these are the mRNAs. They'll get captured by this little gadget that is attached to the bead. I think these are called gems in the 10x case. And they basically capture these RNA molecules. And it's really important for today that they capture them by their poly-A tail. So you have to imagine this little bead. It's got these little appendages that extrude, and it's grabbing the poly-A tails of lots of mRNAs inside a droplet separately from all the other droplets. And if you have a barcode, it's called, which is just a DNA sequence that's the same for all the sequences on one bead, but different between the droplets, then you can, at the end, sequence the product, and you get a paired end read, one end of which represents the barcode, and the other end gives you a little bit of sequence of the end of the gene. So that way you can match up reads that come from RNAs to barcodes that are really telling you which transcripts were in the same cell. So that's the idea, and it's been a popular single cell RNA-Seq is possible with other methods. The technology in general has been popular and transformative, I would say, over the last year or two. So in the case of this technology, this is a cartoon of what's happening here. So really, the previous slide, you had some cells. You run it through this machine, and really, there's all this microfluidics going on. But at the end of the day, you get out what's important, pairs of sequences, barcode for a cell, and a piece of transcript from the 3' end, and you sequence that. Then there's a data analysis pipeline, and this cartoon actually is from software called Cell Ranger that is provided by 10x for doing this for you. And then you look at it. And what everybody agrees with, 10x believes this, but everybody else agrees that the raw data are the gene counts that come out of this whole process. And I'd like to break that down a bit. So out of this whole workflow, raw data is what you have right there. So let me break it down and explain why that is. So this is the whole workflow. Why do we agree as bioinformaticians that the count matrix is the primary object for working with? Well, first of all, if you look, let's ignore for a second the bioinformatics pipeline and just think about the experiment and the sequencing. It used to be actually that part was raw data, but it's not anymore. So back in the old days, 2009, when people were doing Illumina sequencing, they would actually think about the raw data coming off the sequencer, which is actually images. And there's a whole lot of steps to working with those images: analysis, background subtraction, corrections. You then have to base call, you have to phase, crosstalk, all these issues. People wrote papers about them and published them in journals and came to conferences like this to talk about it. But that's agreed upon basically that we don't do that anymore for Illumina. We do that for Oxford Nanopore technology now. That's okay. But at least for 10x, the sequencing is done by Illumina machines. So this part is considered done and closed. Let's look at the pipeline part. So the first step after you get the reads is you align them to a genome or a transcriptome. We heard about that already at this meeting. And we actually heard about this reference, recent paper benchmarking the methods. And actually, there's room for improvement. I believe that. But basically, it's sort of a solved problem, people say, right? I mean, if you look at this chart here and you believe these benchmarks, percent of total bases aligned, the left columns here are default and then T's when you tune the program. And GSnap, NovoAlign, STAR do pretty well. The 10x workflow is based on STAR. So it's a pretty good program. Certainly if you tune a bit, you get good results. And by and large, people accept you align with STAR or you're going to get some reasonable answer. Maybe different aligners disagree a little bit. But especially for this kind of problem where the reads are all 3' to your gene, it's a pretty straightforward alignment problem. So that's basically considered solved. There's another step, which is actually deciding what the barcode sequences really are. And that's not entirely trivial because there are errors in sequencing, so you could get confused and look at a barcode sequence of DNA and think that's actually a new cell, but it's really just you had a sequencing error. And I actually wrote a paper about that. It's on the bioarchive with my student Akshay Tambe recently. But our paper is really aimed at drop-seq, which is another technology, because in the case of 10x, there's a kind of gold list of barcodes. You kind of already know what the real barcodes should be like. And so it's actually pretty straightforward to error correct 10x barcodes. It's not a great research problem. So I'd say people basically agree that you don't need to go and dig into that. Finally, there's the part about getting the gene count. And everybody agrees about that as well, because the data looks like this. This is actually real data. This is an analysis from a 10x data set I'll talk about a little bit later, looking at the distance from the 3' end of the gene and the coverage. And you can see this is actually a 10x data set produced by 10x, and it looks very clean. There's a peak right at 200 is the mode of the distribution. You're not getting very far into the gene. What are you going to do with these reads, really? You just add them up. And so you get a gene count. It all makes a lot of sense. And so the output of all of this is a gene by cell matrix. And that is, I mean, really the standard by which you put the raw, the standard by which you work with what comes out of the experiment. Everything else downstream starts from there. And there's a website put together by Alicia Oshleck. It's a great resource. It lists, I believe, around 140 single-cell RNA-Seq tools. It was already mentioned at this meeting. This is a breakdown. There's visualization, ordering, clustering, differential expression. There are a lot of tools. They all start with the gene by cell matrix. And that's a good thing because having some standards like this means that we can modularize research and workflows so we're not all chugging along in incompatible ways. I want to talk about one of them in particular, differential expression, which is one of the categories in that website. This is a list from another paper, and you don't have to be able to read too clearly here. This is a recent review by Charlotte Sonneson and Mark Robinson, looking at lots of, by lots I mean about 20 or 30 methods for differential expression. I say 20 or 30 because some programs could be run in different modes, so they're really, in a sense, a different method. And what I wanted you to take away from this slide is that there's an input column here, and all the inputs, mostly, let's say, a couple of others, say raw counts, the raw data. Right? So everybody agrees. Right? So that's the raw data. There's some methods that take in things with funny CPM and TPM, and I actually believe that people who do differential expression feel a bit uncomfortable about that. It's like vegetables that have been out for a week. You'd rather have it raw. In terms of the actual methods, there's another recent review of differential expression by Del Molina and others from earlier this year, where they actually did a deeper dive and benchmarked all the methods. This is a plot from their paper. There's a couple of, you can't read this very well. It says MAST, SCD. These are new tools for differential expression, D-Seq. There's some disagreements, but actually there's a set of tools that do pretty well. These folks believe that, if you read their paper, that the slightly better method is SCDE. SCD is a tool designed specifically for differential expression of single-cell RNA-Seq that models this issue of dropout, which is that sometimes, frequently you're missing data in cells. SCD runs, they go into this as well, and I don't want to talk about it in detail, but SCD is slow. It scales super linearly with the number of cells, so it has some disadvantages. But anyway, that's the state of the field. So I thought what I would do is work through an example of how this comes to actually work out in practice. And an instructive data set that's really quite interesting was produced by 10x itself and published in 2016. They have a much larger data set now, but I want to focus on this one. they sequenced 68,000 cells in this experiment. And they're looking at about 15,000 reads per cell. So this is a pretty, you can do the multiplications here. This is a pretty large number of reads, an enormous experiment. And it's sort of a fascinating paper, really. I mean, these are peripheral blood mononuclear cells. So these are peripheral blood cells. And what this picture shows is what's become common in this field. I'm sure you've all at this meeting already seen it, not only at this meeting, but in many other meetings. It's a sort of two-dimensional picture of the cells colored by their type, which is not biologically inferred here, but inferred by clustering just of the transcriptomic profiles. Just to give some detail about how they actually did this, as it will be relevant in a minute, they did something along the lines of taking the 1,000 most variable genes and then looking at the top 50 principal components and doing k-means clustering on those 50 principal components out of the top 1,000 most variable genes. Then they did this thing called TSNE, which is a way to project the data to 2D that has nothing to do with the clustering, but on top of which the clustering is painted. So different cells ended up in different clusters, and they paint them. Now, this part is not standardized. You can already see that because whenever I read a paper like this and I see the number 1,000, for example, picked 1,000 variable genes, I get very uncomfortable because I like prime numbers. So 1,000 is not prime. Neither is 50. So you have a kind of sense that there's some ad hoc choices here. And maybe you did different things. You get different results. And they were very actually careful in this paper. They ran a whole other workflow through a program called Surette, which is becoming a widely used set of tools for clustering and RNA-Seq. And they showed that actually they got something a little different but similar. What's really cool about this particular paper, though, is that unlike most single-cell experiments, they did a sort of form of validation. In addition to just sequencing these peripheral blood cells, they did a whole other set of single-cell experiments where they antibody fact sorted for purified cell types in blood. So there are antibodies for various blood types. They fact sorted them so that they had purified cells of specific types, single cell. And then what they did is they did a sort of alignment where they took each of these cells here, 68,000, and they aligned it to these different profiles for the different cell types to try to label for each of these dots what type of cell is it really. So that's actually kind of cool. And this is what they got out. And I think this is really an exciting moment in genomics. Sort of last year, you can see that you basically can identify by clustering purely bioinformatics. You're just doing some statistics and math and CS. You're actually pulling out cell types in blood. Now, the thing about it is they got NK cells very nicely defined in gray here, and monocytes, dendritic cells. There is, however, a very large globular cluster in the middle where they didn't actually do so well. And they comment that, well, it's not perfect. Just so you really understand here, so again, these purple cells, they have been fact sorted based on an antibody that pulls out the genes CD8 and CD45, RA+, that's actually a specific isoform of CD45. And unfortunately, purple is all over a couple of if you go back here, there was purple and orange were two different informatics clusters, but actually there's purple all over both of them. And yellow is also kind of all over both of them, so that this region here is ill-defined. But I think I agree with them that on the whole, it was a pretty good experiment, even if this particular validation for memory cells and naive cells is not as good. So one interesting thing about this validation is the CD45 antibodies come out of Bio-Rad. They actually target specific isoforms of CD45. So that's actually interesting. And in particular, I wanted to line these up here. So yellow here, the memory T cells, those are selected for by, that's this isoform down here, missing exons 4, 5, and 6, whereas these two cell types, the naive T cells and naive cytotoxic cells, are being selected for RA+, that's up here. That includes exon 4. And so actually, it might not be that surprising that you have this globular cluster here, because really, maybe what sort of distinguishes these cell types here is partly isoform information, but you cannot get to that with 10x. It's not really approachable. In fact, if you look at their data, and we did this, you ask, look, let's take the memory cells and the naive T cells, so those are in purple and blue here, and just look at the counts. These are from the purified cell types. With the 10x G count table, you can't tell them apart. Okay, so you really cannot resolve this specific, the isoforms of this gene with this data. We wanted to be really sure of this result, so my postdoc, Vasilis Dranos, did an analysis where he reran all the data. We're using our workflow. Everybody has their own workflow now for this part. I'll tell you more about it in a second. And he sort of checked carefully. So there's a negative control here, actually, because these two cell types, the naive T cells and the naive cytotoxic cells, are both been selected by the same antibody for the same isoform of CD45, and they show no difference. But the positive controls, where you really would like to see a difference, you don't. You can't, because you're doing gene counts, and that's the end of it. So I'm now ready to tell you the outline of the talk. And really what the outline is, I'm going to show you that actually you can tell these clusters apart. And yeah, sure, there's no differential gene expression, but there is differential transcript usage, and you can find it with 10x data, which is a little bit surprising. That's part two, because first I want to tell you why nobody's actually figured that out. And that has to do with the story of Procrustes, which is the title for my talk, which you've all been waiting to hear, I'm sure. Okay. So let me tell you the story of Procrustes. So Procrustes is a Greek mythological figure, and he had a sort of roadside motel in between Athens and Eleusis, which is actually a road you can travel today. It's 24 minutes by car, but in ancient Greece it was called the Sacred Way. And Procrustes was right around here. There's actually record in Greek mythology that he was on a mountainside right here. Travelers were tired midway. They wanted to take a break. So he offered them a bed to rest in. But the thing about his bed, and he didn't tell his travelers until they agreed to lie down, is that he would fit them to the bed, literally. Okay? So if they were too short, he'd stretch you out. This is horrible torture. But you know, we're about to talk about the bioinformatics soon, so you got to get used to this. If you were too tall, he would chop your legs off. So you'd have to be fit to the size. Now you might think, you know, Procrustes, okay, once in a while, you happen to fit the bed exactly. Well, he had two beds, just in case. All right, so he could swap it out. So he fit everybody to his bed. And, you know, I'd like to argue that what people have actually been doing subconsciously, maybe not so much, is fitting the problem to their method a lot of the time rather than the other way around. Essentially, they're being Procrustean. Particularly, if you look at a lot of these methods, some of these methods in here, like D-seq, EdgeR, they were not even developed for single-cell RNA-Seq. They were developed for bulk RNA-Seq. Obviously, the nature of the data is completely different. There are a lot of, you know, when you're trying to compare cell clusters and figure out which genes are different, there are a lot of cells versus bulk RNA-Seq. Usually, there are very few replicates. There are dropouts. There are a lot of things. But it's convenient to just take the same raw count input. People look at this pipeline, and I've done this myself. You really don't feel like going back into the real raw data. I taught a course once where we had the students actually download the images that Illumina machines put out. It's not fun. It's not, you don't want to go there. So people really don't want to go there, and it's just convenient and comfortable to stick everybody in your little Procrustes bed. I think the bed looks a little bit like this. It's a sort of California king size, you might say. It fits maybe a pair, particularly marginalization and projection, which I'll talk about. It's very frilly and decorated. You know, there's normalizing the data up here. It's very important. There's, the license for the software down here. All this is very, very important. But you just stick the data and you put it in your bed and you chop the legs off. Right? There are really two issues I want to talk about that people have fixated on, and really all of those 20, 30 methods do the same thing. What I mean by marginalization is that reads are aggregated to raw gene counts. That just goes without saying. And that's marginalizing because you're just summing up. Okay? So it's a sort of form of averaging. There's a second issue, projection, which is you fixate on one coordinate at a time. So if you're looking at genes, you test gene by gene. Sometimes people are doing transcript differential expression, not for 10x data, but in bulk RNA-Seq, for example, or even with some single-cell methods that give reads throughout the transcript. Then you fixate on one transcript at a time. Every single method does one of those two things. And that's a sort of way of simplifying your data, because marginalizing reduces your dimension, really, and then projection tests one coordinate at a time. So that's the Procrustes bed. So I'm going to begin with marginalization and talk about why it's not necessary for 10x and how you get around actually doing that. And I'd like to begin by telling you that I've told you a little lie. And it's OK, because I'm confessing. I showed you the 3' UTR distribution of 10x data. Now, this was an analysis put together by my colleague Paul Melstead. It's actually a non-trivial analysis. What he did to do this is he aligned the 10x data to the transcriptome and ran a quantifier to calculate the posterior probability for each read to be in each transcript and where in each transcript. And then plotting here the expected number of reads at a distance from each transcript on average. So that's sort of how this was made. And I showed you this picture. I actually cut off the axis there. So this is a bit of a tutorial how to prepare your future talks. So if you actually zoom out a little bit, it actually reads all the way out to 5,000. Maybe it looks like, so this is, decreasing. There's a little bump over there. You can't really tell so well, so I did a log plot where this is the log counts on the y-axis. And there's actually, this distance from the 3' end, we're at 10,000. In fact, 500 was over there. 20% of the reads are more than 500 base pairs away from the 3' end of the transcript. Now, that should actually kind of surprise you because at least, honestly, until Paul made this plot, I really believed that the image in my mind was 10x. There's a barcode, and you grab something off the 3' end. It's close to the end. It's really, that's it. You're not going to get stuff 10,000 base pairs into your transcript. But actually, that's what we find, and I'll show you why in a minute. This is sort of a picture of the CD45 gene in IGV browser. It's a little hard to read. I'm showing you a big gene. It's called PTPRC, and this is just a part of it, actually. There are lots of isoforms. You know that already because you saw that there are antibodies targeting some of these isoforms, but there's a lot more isoforms than were on that Bio-Rad site. Some of them have 3' UTRs in the middle. And actually, we pick up reads. There's a little bit of reads right there. But we also pick up reads where there's no annotated 3' UTR, probably because we don't know all the 3' UTRs. But it doesn't mean you didn't get a read from somewhere in that transcript. There are actually reads all over this gene. This particular figure actually was made for this project, basically. To do it, Paul actually implemented a new feature in Callisto. Callisto is our RNA-Seq quantification tool. This is a feature many people have wanted it for a long time. Callisto does what's called pseudo-alignment. I'll discuss that also in a little bit, but it's been not possible to visualize what comes out of the program. We have had pseudo-BAM for a while, but it produced BAM files which were in transcriptomic coordinates, and now we can actually directly output genomic coordinates. And Paul has poster 131 right after this, where he'll talk about that and show some really cool things you can do with this technology. One of the great things is it's great to view things on IGV. Usually, it takes forever to do an alignment. This data set here had 3 purified cell types you're looking at. And it was close to more than 500 million reads, I think more than 600 million reads. Takes us a couple of minutes to process data set. And during the course of this project, we kept reprocessing this data in different ways over and over. You can continuously visualize it. So that's what makes this feature really cool. By the way, we looked at other annotations, not just ensemble. This is actually the gene set from chess, which was described at this meeting. It's a really great project, which I think is long overdue, to really try to annotate the human genome properly with all the RNA-Seq we have. This is Michaela Perteo's project. But even there, most of these 3' UTRs are missing, and we have reads all over this transcript with no functionally annotated 3' UTR there. So we are a long way off from getting these 3' UTRs, but it doesn't matter. We can still find the reads on our existing transcripts. But we have a problem. We'd like to do some sort of isoform level quantification here, but how? Every method that will do RNA-seq quantification at the isoform level requires, at a minimum, some sort of a model for redistribution across the transcript, usually uniform model. Even if you don't assume uniform coverage of reads, which is certainly not the case here, at best you have some model of a decay of reads, but you can see here that every single gene, I'm not showing you others here, you take my word for it, but you can see it just from this one, has some completely aberrant, unique ad hoc pattern that's going to be different from another gene. You can make this averaged plot that I showed you before, but it's not going to be very informative for quantifying this one gene. There's not much you can really do, and people have just been adding up the counts. So the insight, actually, for how to deal with this came from my former student, Cole Trapnell, and from an interesting dinner we had a little while ago in Berkeley. Cole actually is the developer of cufflinks. As you may know, if you've used cufflinks, we were advocates from the outset for doing transcript level analysis, and Cole and I have been advocates of that, and I've preached that on this very podium. But Cole is now an assistant professor at University of Washington in Seattle, and he's gone into the wet lab. And he sort of whispered to me and said, you know, Lior, I've realized my biology colleagues, they're not interested in transcripts. Don't tell anybody, we want citations. Okay. And Cole was really forthright about this and explained to me something I just had never thought of, because I had not done experiments in my lab at that point, which is that it's very hard. It's not that transcripts are not important or that biologists don't believe that, but it's very hard to test a prediction about an isoform. It's difficult to knock out an isoform. Maybe now with CRISPR-Cas9 you can go and knock out an isoform. But really, it's very hard usually to target individual isoforms. That CD45 antibody is a special case, but usually it's very hard to do anything with the result. So Cole was saying, you know, we need to kind of think about how to reconcile. We don't want to not look at isoforms. It's more powerful to see what's happening underneath the hood of the gene. We're not, you know, we believe that, but maybe it's not so good to really just pretend that that output is really useful. At the same time, another former student of mine, Nicholas Bray, got a PhD in math, also went into the wet lab. I'm trying to figure out if this is a good thing or a bad thing that all my students are going to wet lab. I don't know. They're all going that way. Nick works for the IGI. He's really an experimental biologist now, but because of his bioinformatics background, he's sort of the lab, the go-to guy for compile questions. And he said the same thing. You know, my colleagues, they want gene-level DE, sort of a transcript-level resolution. We heard a beautiful talk yesterday about isoform switching in cancer. That's what biologists want. The isoform switch, they want to know something happened in the gene, but they don't necessarily care to know the details of the isoforms. So this got us thinking about how to do this. And my student, Lin Yi, is a Caltech MD PhD student, was looking at, you know, cases like this in our data. This is actually an experiment, a bulk RNA-Seq, which I won't talk about today, published earlier this year. There are two conditions, red and blue. This is an example of isoform switching. I mean, this happens all over the data we've looked at over the years. On the right-hand side, you see that at the gene level, you don't see any difference. How do you like do an analysis at the gene level? And the idea we came up with is really straightforward, is to sort of think about this as a meta-analysis problem. Instead of first aggregating reads and then going on to analyze, which is what people typically do when you get gene counts. First, you'll do analysis and then aggregate. Now, I'm not talking about single cell right now. In the bulk case, that's a very tractable proposition. What we did is, instead of first quantifying gene abundances, first you calculate p-values for individual isoforms, like we were doing already in cufflinks in 2010. Then you will take those p-values, and rather than reporting them, you will combine them by gene. How do you do that? Well, under a null hypothesis, where nothing is changing in the gene, those p-values should be uniformly distributed. And there's a long history in statistics, starting with Fisher, to test whether multiple p-values that came out of multiple studies are in fact uniformly distributed. It's a simple thing. But actually, out of all of Fisher's methods, this is the one Fisher's method that actually most people don't know so much about. This is very popular when people are doing meta-analysis where they've done different studies, let's say in cancer, and they want to combine all the data. That's usually where this is used. It is used a lot in GWAS, this kind of idea, not so much in RNA-Seq. There are lots of different ways of combining p-values, and Lin tested a very specific one, which is called Lancaster's method, which is a modification of Fisher's method. I'll just tell you what it is. It's really straightforward. We get the p-value using Sleuth. Sleuth is a new differential expression program for RNA-Seq that's specifically targeted to getting transcript-level p-values in bulk RNA-Seq settings. What you do is you take your p-values and you sum up twice their log. And the negative of that, under the null hypothesis that those p-values are uniformly distributed, should be chi-squared with 2k degrees of freedom if you combined k p-values, so you could go test whether actually they're chi-squared or not. And that gives you a new p-value for your p-values. For example, if you combine two p-values, actually this should say k, I apologize, then this is the formula you use to combine them. And you can plug it in. If you put in 0.09 twice to this formula, you get 0.05. So you're going to have two transcripts that are not significant in and of themselves, but together actually indicate there's something going on in the gene that is significant. Once you get the gene p-values that come out of the aggregated p-values now, not the aggregated counts, you can go and plug them into a standard multiple hypothesis testing correction. And this way, you gain power by virtue of not doing tons and tons and tons of tests on transcripts. So this is one way to get around the marginalization issue, at least for bulk RNA-Seq. And actually, if you do that in this example, you can't read it down here. There's a p-value that's slightly higher than this one. It's 0.0019. Together, the whole is greater than the sum of the parts. You benefit from the reduced correction in combining p-values. We did a simulation study, and we showed that compared to native sleuth and also actually any other method you might try, there's a tremendous increase in sensitivity of such a method by virtue of pooling together everything under the hood. So this is a gene-level plot where you're looking at false discovery rate and sensitivity, and green is this Lancaster or Fisher's method. Blue is another method here that's sort of alternative based on taking the minimum p-value, not as good. So how about 10x data? I mean, it's all nice to sort of do away with getting gene counts when you can do things at the isoform level. But you can't do that with 10x data that has this very strange distribution. And so Lin had this idea, which is to say, look, let's break the bed. Let's ask what's upstream of quantification. What are we doing to get the transcript quantifications? And well, what's upstream of transcript quantification is read alignment. So I don't have to talk about read alignment here. Usually you have reads, you have transcripts, and you find out where they go. But that's not really useful for doing something like differential expression. So what's upstream of read alignment? And what's upstream of read alignment is pseudo-alignment. And Lin knows about pseudo-alignment and has thought a lot about it because in my group, we talk a lot about pseudo-alignment, having developed Callista last year. In pseudo-alignment, you're not even going to the read alignment. You're simply looking and computing a table like this, where for each read, you simply ask, is it in transcript 1? Is it in transcript 2? Is it in transcript 3? If it's in transcript 1 and 3, like read 1 over here, well, you give one count to the set T1, T3. And so what comes out of pseudo-alignment is really upstream of even alignment, where you're just measuring the multiplicities of how many times you saw different sets of transcripts in your data. That's all. It's a very crude level. But what Lin realized is that, counts is great. Everybody wants raw counts. Well, these are counts. Let's plug them into something. It's a strange thing to do, because these things don't mean anything biologically. They just are bioinformatic constructs that come out of the ambiguity in read alignment. But they produce counts, and in different experiments, you can ask whether these counts are actually different. So we have a tool for producing the actual pseudo-alignment counts. And we actually, I'll just go back for 1 second, I'll say that we have terminology for this. The sets we call equivalence classes, but the counts, we call them transcript compatibility counts, or TCCs. And Callisto, which is usually, I think, used to quantify isoforms, actually has a mode, Callisto pseudo, which you can run also on single-cell RNA-Seq, and will simply produce these sets with these counts. You get a matrix where the rows are labeled by sets, columns by cells, and you get these integer counts. And you can read a little bit about how to do that here. So what Lin set out to do is aggregate p-values from comparisons of TCCs, of these weird counts that don't mean anything biologically, but are actually proxies for what the transcripts are doing underneath the hood. And I'm really pleased with this figure. This is the same data set I showed you before. Not sure what's happening there, sorry. This is this data set, 2 conditions. On the right-hand side here is where the analysis where we quantify transcripts. Let me try to fix this, because this is going to be not so great. Okay, it's good. So on the right-hand side is the more standard analysis where we quantify transcripts and combine their p-values. On the left-hand side is the analysis where we just used these TCCs. And these are the top genes on the list. And I've sort of put red bars so that you can see they're a little bit out of order. But basically, what this analysis produced, this analysis produced. But it's kind of it takes a while to wrap your mind around this. These genes have been identified as differential between 2 samples without ever quantifying the data. There's no gene counts. There's no transcript counts. We don't know which gene is more highly or less expressed than another. We've simply taken the counts of these sets of reads that display the ambiguity in the alignments. So motivated with this idea, we thought, aha, you can do TCCs on bulk. Well, let's try that on single-cell 3' data like 10x. But there's one other thing here, which is that there are a lot of TCCs, and they're not independent. So I swept something under the rug with meta-analysis, which is that when you use something like Fisher's method, you're assuming that the p-values, not only are they uniformly distributed, but they're independent. And that's not true when you generate counts for these sets, because if 2 sets share some set of transcripts, it's clear that the counts you're going to get for them are not independent. And so projection, in this case, meaning test each TCC to get a p-value, is not such a good idea and is problematic. And that's where Vasilis came in and had a really, really clever idea that's very simple but really non-trivial. If you look at the literature, and this is a recent paper from 2016, but if you go back all the way to 1990s, late 1990s, when people were doing microarrays, people wrote about logistic regression. Now, everybody knows logistic regression. It's not a very popular method for differential expression, and it's the microarray guys back in the late 90s that ruined it for us. Why? Because there were not enough replicates. To do something like logistic regression, you need lots and lots of replicates. So the number of genes far exceeds the number of samples, so logistic regression has been abandoned. I mean, it's really not a method that's mainstream. Vasilis suddenly had this epiphany that in single-cell RNA-Seq 2017, one of the things that those methods, there's 20 on that list that I showed you in that review, they're ignoring the fact that in a cell cluster, when he's looking at 68,000 cells, you have hundreds, sometimes thousands of cells in each cluster. it's essentially as if you did a ton of replicates of your cell type, and you measured the variance. You can measure it very accurately. So Bacillus had the idea of doing logistic regression with this kind of information. What's nice about logistic regression is it's essentially an unsupervised approach. You're not making any assumptions about the TCCs or the isoform switching or any of that stuff. This is what your picture looks like. This is one gene. This is a conceptual figure. You have two TCCs counts, or two equivalence classes, I should say. So you have two sets of transcripts here, and you're just measuring the counts for them. And you have two cell types here you're trying to do DE on. And so the red cells and the blue cells, that's what you're trying to tell apart by doing differential expression. What logistic regression accomplishes is to find a line that sort of best separates out these two point sets. If they happen to lie like this, it'll find a line that's at that angle. If you have isoform switching, that means that one TCC goes up while the other goes down. This line will be rotated 90 degrees. Essentially, this method, it will go searching for the best hypothesis for you to test to identify DE in your gene. It's a really good idea. So we've tested this idea. It's really like, it has to beat other methods. By the way, to do this, Lynn had to develop her own simulation framework. Because of the Procrustean bed, there isn't a single simulator that I know of for single-cell RNA-Seq, despite there being 140 methods, that will simulate reads. They all simulate matrices of counts. But to do something like this, you need reads, so Lynn actually developed, along the way, her whole simulator. In blue here, you have simply logistic regression on the raw counts. We've tested what happens when you do different normalizations, and you can actually do a little bit better than this. But basically, this is a native function in R. You don't even need a packet. Logistic regression is a native function in R. And we actually do find that SCD slightly outperforms other methods. So, and I won't get. We have our simulation is a little bit like along the lines of splatter. We try to mimic the features of single-cell RNA-Seq. We learn how to do this from a real data set. I think it's a pretty reasonable simulation. We're actually not even simulating anything from a model here that has anything to do with us. SCD is a little bit better. It's very, very slow. So let me go back, and I think I have just a couple of minutes left, and I'll complete the circle, if you like. I showed you that at the gene level, the CD45 isoforms were undistinguishable, or the CD45 gene, I should say, was indistinguishable. This is what things look like when you look at the TCCs. So there are about 8 equivalence classes that have non-trivial set of counts, and they change quite a bit between conditions. We don't know anything about what they are. They're just some numbers, but they correspond to sets of transcripts. All right? When you combine all of this information by logistic regression, you get a very significant p-value for this gene. We add another positive control. Remember, there are actually 3 purified samples here, 2 that's selected with the same CD45 antibody, and 1 with a different one. So the second positive control comes out very nicely significant, and we have 1 negative control. Again, these are 2 totally different 10x runs. We're not even normalizing anything, nothing. We get nothing significant on the negative control at the isoform level because the isoforms are behaving the same way. So at this point, I have to admit that I myself am Procrustean. So we developed Callisto already more than 2 years ago, and it's something people have frequently asked us about is please help us visualize these kind of things, equivalence classes. And we haven't done it because, well, we're all busy. It's actually a really non-trivial thing to couple together all these sets of transcripts with the coordinates of where the pseudo-alignments are and all this stuff. It's a little bit lazy and Procrustean, but we were really, really curious now, what are these TCCs, and do they correspond to the actual isoforms that these antibodies target? So Paul Melstead went back and added a new feature as well, which is to actually output a bed file that shows the TCCs. And I'm sorry you can't read the numbers here, but each one of these smudges here actually is an equivalence class. So what you see here is, let me go, for example, here, these black bars here are locations in the genome where reads that align to those locations are ambiguous in the same way to transcripts. So you can see that the equivalence classes are highly non-trivial. Callisto is identifying these naturally from the data. And what's differential are these sets, right? We're finding different counts for these different sets. This is a sashimi plot showing the counts of junction reads. And what I wanted to show you is, actually I think I have two of them. I'll show you this one. It's a bit cleaner. In this plot here, this is exon 4. And you can go and verify that now by looking at the gene structure. This is the antibody map from Bio-Rad. You can see that 4 is absent in memory cells and present in the naive cells. So this is RA and this is RO. And this little bar right there, that's the equivalence class. That's right there. It's the little one. It doesn't look like a lot, but actually of all these equivalence classes, it has the highest fold change. It's got a lower read count, but it has the highest fold change. And it was a really exciting moment to go and map each of these to the different transcripts. And you can find some of them are from UTRs adjacent to these skipping events. Some of them correspond to junction reads. I believe this big one right here is from a UTR right around exon 6. And it's really 4, 5, and 6. And they're deeply embedded far into the gene. So final thing I want to say in the last remaining few minutes is we thought, let's go back into the real data set and see if we could have actually found this de novo. Because in already last year, Vasilis wrote another paper where he actually argued that equivalence classes are also really good to cluster on. We really believe philosophically that you should do away with gene counts and just do your whole bioinformatics with these counts, these TCCs. And so here Vasilis clustered the actual 68,000 cells based on these transcript compatibility counts, the equivalence class counts, without ever quantifying genes. And we also have a clustering method, but this is a standard clustering method, Lavane clustering. It's not our own clustering. And he pulled out a whole bunch of set of clusters. You cannot pull these clusters out with gene level analysis. He then mimicked the 10x analysis, taking the purified cells and the cell markers. There are other cell markers, I didn't go into this, for these cell types, and found that clusters 1 and 2 correspond to naive T cells, cluster 3 corresponds to naive cytotoxic cells, and cluster 6 corresponds to memory cells. This is the breakdown of all of those 6. Reproduced these plots for those same equivalence classes. So on the right-hand side here are what I showed you previously, the purified cell types. But now we're doing the same analysis de novo from the 68,000 peripheral blood cells. But simply having identified the clusters with the power to resolve isoform differences because we didn't toss away the baby with the bathwater. It's really pretty cool. You can see that many of the equivalence classes display the same changes, really. There are some differences. Here you see a change that's a little bit different there, but after all, we haven't purified cell types here. So really, this 10x data has in it these cell types in a way that's just not visible if you do the collapsing or marginalizing. So I have 2 take-home messages, and I have 2 minutes. I won't spend 2 minutes on them, but I'll finish with these 2 messages. The first is that I'm proposing a new workflow for single-cell RNA-Seq, which is that you start your raw data is this TCC matrix of equivalence classes by cells. It's much easier to produce than the gene count matrix. You get it out of a pseudo-alignment program. You don't even need to align your reads. It's much, much faster to get this matrix. It's much simpler. And you can do all the standard things you did with gene counts, normalizing the data, all that stuff. You can do it with this matrix. But it's just a better representation of the data. It's more useful. It has a lot more information in it. It means going to those 140 tools and replacing their input. And that's really a big deal. It's a lot of work, but I think we should do it. We also, I don't have time to talk about it today, I also have a view of changing the output. one of the things that came out of this analysis is that we sometimes have equivalence classes that cross between different genes. And then we cannot just look at a gene in isolation. So what we do then is we actually look at sets of genes, and we find that they are differential as sets. We basically take the connected components of this graph. And we found all sorts of very interesting stuff in this blood sample in gene families that are dead from the outset when you just collapse by gene and look at unique counts that way. So I think that the output should not consist of single genes, but on gene sets, which is a new way of thinking about what a differential expression analysis actually produces. I should also say that we ran this method on the data, and we found it's not just this gene that switches isoforms where you can find this. We have at least a few dozen genes which are flat at the gene level where we find differential expression. The second take-home message is mind the mean. It's convenient to average in genomics. I wrote a paper a couple of years ago with my former student, Maramit Singer, on a setting in DNA methylation analysis where this Simpson's paradox having to do with averaging has led to false conclusions about differences in methylation between functional genomic regions. It's dangerous to average, and it's often not necessary. It's just convenient, and we shouldn't be lazy. We shouldn't do it. We're partly not to blame. I've pulled out the culprit. As far as I know, the original sin, this is the figure. It's figure 25 in the mouse genome paper. They made this really cool plot where they plotted the average conservation. This is the metagene. You collapse conservation across all genes by sort of averaging the conservation. There are indels in these alignments, and they present a big problem here, which is what we discussed in this paper. Interest of full disclosure, I'm an author on this paper. So there you have it. So I'll just conclude by thanking, first of all, the Genome Informatics meeting, not just this year, but all the years I've come here. Really love this meeting for its focus on methods. I'd like to thank Janet and Melissa and Aaron for inviting me and hosting, I think, the largest meeting yet and a really successful meeting so far. The project I discussed is the work of primarily Vasilis Dronos, who had these ideas, Lynn, my student, and Paul. I've mentioned most of these students. Jace has been key. He's a molecular biologist in my lab, and it's been really key in having us think through a lot of also the molecular biology, but the bioinformatics in our lab, and identify places where we are averaging and where we really need to reconsider our workflows. Finally, I've just moved to Caltech. Some of the work was done there at Berkeley, and I'll thank the NIH for funding me. Thank you very much.
So with the interest of the poster session, we'll take just a few questions right now. One in the back, and we'll get the one here first.
Very nice, Lior.
Thanks.
I'm wondering about the origins of these reads in the interior of genes. Are they priming from poly A sequences in the middle? Is it random priming? Is it something else?
That's a good question. We actually have looked at this in the browser, and I believe they're unannotated 3' UTRs. And we can see that because we can see some of the poly A sequences bleeding off the reads into those regions. So they're just, I think, alternative 3' UTRs. And we have the evidence for it. It's actually, yeah. So.
Great talk. My question is the new pipeline with TCCs,
yes.
Is there a normalizing step, and how does that work?
We've actually looked at that. So in the results I showed today, we didn't do any normalization on the counts. But we've also explored normalization. We kind of like the DE-seq2 normalization so far. We'd like to explore it more, so I didn't talk about it too much. But I think that they should be normalized, obviously, by the total number of counts. In this specific analysis, we didn't normalize because 10x was very careful in sequencing the same number of reads and the same number of per cell and the same number of cells. I should say, to really be careful, we downsampled to 3,000 cells for each of the data sets when we did the logistic regression. So yeah, I think one should normalize, and I think the standard methods people have developed will apply and look good, at least in our simulations.
Great talk, thank you. So question, you couldn't do the Fisher's method correction on the counts from all the TCCs because there's a correlation structure. But there are modifications on the standard meta-analysis that account for that correlation structure, which you could, in theory, explicitly compute because you know the correlation between the TCCs. Did you look into that?
So that's a great question. First, I will say that we actually found that just applying Fisher's method naively on the TCCs works really well. It's not that bad. It's better to do logistic regression, but it's not bad, actually, and we've shown also with bulk RNA-Seq you can do very well. Very similar to transcript level analysis. It is true, and I'm very interested to figure out how to deal with the correlation structure. The problem is that it's not so easy to actually determine that correlation structure because it is somehow data dependent. The TCCs themselves are data dependent, and the correlation structure is going to depend on the abundance of the constituent isoforms that make up those TCCs. And so I have some ideas of how to do it, and I'm very interested in that, but I don't quite know how to do it right yet. So.
All right. So we'll have the poster session, and then at 7 is cocktails, and at 7:45 is dinner. Please don't skip the posters. They're really fantastic. And let's thank Lior again.
Thank you.